Mm.
Cool. Well, welcome everyone to our August edition of the Coast to Coral online seminars. Before we begin, I would just like to pay my respects to the traditional owners of the lands on which I live and work and all the lands on which everyone's gathering tonight virtually. And I pay my respects to their elders, past, present, and emerging.
Ilya, we're not seeing your slides moving.
Oh, you're not?
No,
Oh,
we've still
great.
got the first slide.
How's that?
No.
No.
Maybe you need to stop sharing and start again. Oh,
Sorry, everyone. All good?
that's better. Now we've got the second slide.
Okay, great.
Yes, we're all
Right,
good.
so I'm sure everyone is aware that tonight we're going to be introducing PJ for her talk about tiger sharks. But before we get right into tonight's online event, I just wanted to give a bit of a brief overview about Reef Check, who we are, what we do, and then we can get into the actual seminar portion of tonight's session. So for anyone that doesn't know, Reef Check Australia is a not-for-profit organisation, and really we're about protecting our local reefs and empowering other people in the community to do so as well. And we do this mainly through hands-on research and education, as well as our long-term monitoring program and community events. Sorry, my slides are not moving. Great. So just some housekeeping before we get started. Obviously, just general online etiquette. Please feel free to keep your videos on while there's quite a few of us here, but while PJ is talking, of course, keep yourselves on mute and avoid any sort of background noise or chatter. If you're not on our mailing list yet, we've just got the SEQ surveys email up on this slide. Feel free to get on our mailing list. We will send out information about future seminars that we host, usually on the first Tuesday of every month, as well as any of the other events that we run. Mainly, it'll be about the Southeast Queensland events. So for the Brisbane, Sunshine Coast, and Gold Coast regions. I'll also probably take a little group photo towards the end of tonight once more people have sort of joined on. So we'll just skip that for now. So our upcoming events, we've got the Gold Coast Whale Festival this weekend. So I believe that's on Saturday. It'll be at Justin's Park in Burleigh. And we actually have a stall set up there with a bunch of our ambassadors that'll be around, and it should be a really great community event. It sounds like there'll be researchers from various universities and organizations. I believe SeaWorld and some other institutes and groups will be present as well. So it's sure to be a lot of fun. There's also the Plastic Free Sea Festival. We'll also be hosting a stall there. I believe it's being hosted by the Visionary Ocean Warriors and a couple other groups. That will be in Caloundra, so for anyone on the Sunshine Coast. And it'll be in the morning of Saturday, the 23rd of September. There's also the Caloundra Music Festival, so another event for our Sunshine Coast volunteers or community groups. They're over the last weekend of September, so Friday the 29th through to Sunday the 1st of October. And I just want to reiterate, you don't need to remember all those dates I just mentioned. A lot of these events will be posted on our mailing list, for example, but also on many of our other social media platforms like Facebook and Instagram. So I also just wanted to use this time to highlight our event next month, our next Coast to Coral online edition, which will be by Madison Brown, who's based at the University of Queensland in Brisbane, and she'll be talking a little bit about restoration opportunities in a UNESCO biosphere. And I believe she works a lot in the blue carbon space, so it'll be a really interesting talk and a bit of a change. So again, that'll be at 6:30 p.m. online. So keep an eye out for the event and the link to register and get the Zoom link. That will be on the first Tuesday of September, so the 5th of September. And of course, if anyone here tonight hasn't already gotten involved in Reef Check events, feel free to reach out to us if you want to find out more. So you can find out all of our information on Facebook, Instagram, Twitter, LinkedIn, all of those links. So please like, follow, learn, and share. I'd also like to take a moment to thank all of our various supporting organizations and all the volunteer groups that assist in all of our activities. So namely the city councils across Southeast Queensland, Port of Brisbane, and the Great Barrier Reef Foundation. And of course, I'd like to thank in particular the group of people that help with this Coastal Coral seminar series. It takes a lot of work and behind the scenes and getting speakers organized and getting slides and advertising materials out. So without these people, it really wouldn't be possible. And of course, to our lovely speakers, such as PJ, who's joining us tonight to talk to us about her PhD work on tiger sharks. So she's based at the University of the Sunshine Coast here in Southeast Queensland. And she's conducting her research on the biology and ecology of tiger sharks along this Southeast Queensland region. So she's focusing on small and large scale movements and diet, using multiple tagging technologies and metabarcoding to sort of better understand and inform the knowledge gaps in the literature. So this work obviously has some important implications for conservation of this particular species and for understanding the ecological role that it fills, particularly towards providing updated and informed advice for policy and management, and in protection and safety programs for beachgoers. So with that, I'd like to allow PJ to start sharing her screen and get started with the talk, and I will stop sharing. And I will just mention briefly that we'll have plenty of time at the end of today's talk for questions and so on.
And you can also pop them in the chat just in case you think you might forget by the end of the talk.
Well, thank you for that. Hi, everyone. I hope you can hear me well. I'm PJ. Sorry about the blurriness of my screen, but the slides should be okay. So I'll start sharing my screen. Cool. Can everyone see that? Am I good?
Yeah, that's all good.
Okay, awesome. Where is it? Cool. So yeah, just as Ilya said, I'm doing my PhD up on the Sunshine Coast in Queensland, looking at tiger shark, fine-scale habitat use, long-term migratory movement, and diet. I'm trying to understand more about the biology and ecology of such an important apex predator along our eastern coast. So I work with Bonnie Holmes and Chris Clemente and Alexis Levingood up here on the coast. And then I also work with Johan Gustafsson down at Griffith and Paul Butcher down at DPI in New South Wales. So it's a pretty big team and everyone's incredible. Oh, there we go. I'd just like to acknowledge the traditional owners of the land on which we meet for this meeting, for all of my research, for the university. I work along the entire eastern coast of Australia. So just acknowledging the large amount of land and ocean that I work within. So we all know that sharks and elasmobranchs have been around for millions of years. They're extremely diverse. They have worldwide distribution. We have sharks that travel great distances, sharks that remain pretty resident in specific spots, all different morphologies, physiologies, and behaviors to fill different niches in our oceans. But what they all have in common is that they're all incredibly important for our ecosystems. And as more research is coming out in the last couple of decades, we're seeing how important they actually are for maintaining a lot of the marine balance that we're seeing. So Just a little bit of background. Sharks, as I mentioned, extremely important. They are being considered some of the top ecosystem engineers in many of the habitats that they exist in. They provide critical structure via indirect and direct risk effects. And this helps with the different functioning services of these marine environments. So direct effects would be mortality, various predation events, and indirect effects would be more so how the presence or absence of sharks influences behavior of other species. So a really good example is in WA. The presence of tiger sharks keeps the dugong population down and helps to maintain seagrass habitat out in WA. Their populations globally are declining, as most of us probably know. They're vulnerable, many are threatened. The number of vulnerable and threatened species has doubled in the last couple of years. And the IUCN Shark Specialist Group has them down with a 37% increase in their vulnerability. So most of these threats are fishing and climate change related. And this is generally for sharks and also for tiger sharks. Fishing and climate change are having some big impacts on their distribution. And then the elephant in the room, climate change, particularly with our marine environments, we're seeing an increase in ocean temperature, and tiger sharks tend to follow a thermocline for their migrations. So we're seeing a redistribution of their movement patterns, and we're seeing them move further south and staying further south for longer. So part of my research is trying to figure out where they're going to be heading with this change. For tiger sharks, we're having a habitat expansion, but for other shark species, this could also mean a habitat shrinkage rather than expansion. So extremely important and different depending on the shark species we're looking at. Specifically for tiger sharks, they are a highly mobile predator. They've been tracked traveling thousands of kilometers, going down to almost 1,000 meters deep. Females tend to be larger than males, reaching maturity at around that 2.8 meter mark. Males reach it slightly sooner than that. If you look at that, this first picture on the left, you can see that they have a circumglobal distribution in warm tropical and temperate waters. And then on the right here, we have Australia. And my focus is on that eastern edge, looking at the Coral Sea and the temperate east marine regions in terms of movement for this species. Some other facts, they're diverse generalist feeders. They eat, they've been seen to eat 8 of 11 functional groups. Very diverse, very generalist, very opportunistic feeders. And then the tiger shark specifically is listed as near threatened on the IUCN Red List. This varies depending on the population you're looking at, but near threatened is accurate for our population here in the Indo-Pacific. So with the importance of tiger sharks to marine ecosystems, it's highly important that we understand their movements, their behaviors, and what their interactions with both other species and the environment looks like. There was a study that came out in 2017 that simulated the removal of various shark species from a reef environment, and tiger sharks were associated with the most change in reef structure and animal assemblage within that habitat. So they are considered, especially once they're mature, to be top apex predators and ecosystem engineers, quite keystone species for these environments. And to look at how the impact of these environments, we're looking at, as I said before, the direct and indirect effects, because these could potentially be an environmental indicator. And then lastly, we also want to look at diet. So diet obviously is how animals gain energy. Energy is how they're able to move. So understanding where and when they get their energy will help us further understand the different drivers that affect their movement, whether it's long migrations, residency, or something in between. Yeah, so my PhD project is specifically looking at 4 different areas. I'm focusing on quantifying movements of mature male tiger sharks using satellite tags. I'm looking at inter and intraspecies interactions using physiological sensors along with video cameras. And then I'm looking to identify the diet assemblage of these species using 2 different methods, stomach content analysis and eDNA. For this presentation, I've decided just to focus on my first chapter. There's quite a bit that I'm doing, so easily will answer questions afterwards, but I'll just focus on these more long-term, large-scale movements of mature males for this presentation. As I said before, my study area is the eastern coast of Australia, from Hervey Bay in southeast Queensland, all the way down to Shell Harbour in New South Wales, also going out into this, out further east, heading into the ocean and trying to see where they're going out towards New Caledonia. I use the New South Wales contractors and Queensland contractors to help me with my work. So those are just a couple of the locations that I use quite frequently for my tagging. So using satellite tags to track large-scale movement has been done before on tiger sharks, but what we've seen is that most of the tagging has occurred on adult females or juveniles, male or female. And we don't have much information on mature male tiger sharks. This could be due to a variety of different reasons, potentially sex-biased movement, potentially whereas we see mass migrations with females, males might be more resident. We're not quite sure, and that's the focus of my study. I will be focusing on targeting large mature males off the coast of New South Wales, as evidence from game fishing tournaments and competitions over the past couple of years have indicated that that's where they might reside or spend large amounts of time. So by tracking mature males, we hope to identify and quantify large-scale movements and see if we do have any sex bias differences. So I'll be looking at where mature males are traveling along the eastern coast, what type of movement characterizes these migrations, and what the ecological and biological factors are driving those movement patterns. So I'm using 2 different types of satellite tags for this project. I'm using PSATs, or pop-up satellite archival tags. These are attached at the base of the dorsal fin, as you can see in that first photo right here. And what these do is collect information on water temperature, depth, and light-based geolocation for animals that spend time underwater, away from the surface. So I'm able to collect information on their whereabouts without having to physically see the animal. After a set period of time, ours are set for 180 days, this tag will detach from the shark, float to the surface. Once it floats to the surface, the satellite connects with the Argo system, and we get all of the information that's been collected throughout its deployment sent through to our computers, and we're able to have a look at their movements. And I have a picture of what that looks like in a couple of slides. The second tag that I'm using is a splash tag. It's an updated spot tag, which stands for Smart Position and Temperature Transmitting Tag. Essentially, from these, we're able to get near real-time locational data on a tagged individual. But this only occurs when they're at the surface swimming around with the sensor out of the water. So for tiger sharks, it works really well. We've seen that they do tend to spend a decent amount of time at the surface. So we're able to get some really good near real-time tracking information. These splash tags also record depth and temperature. So we get to also compare that with our PSATs and see, collate those 2 different tag types to get a better picture of where they're moving. To do this, I get to go fishing off of boats. It's quite fun. We hook the shark, we get it alongside the boat, and I'm able to, in this position, maneuver it to have its dorsal fin up, and I'm able to tag the shark. At the same time, I'm able to take my anal swabs, which are part of my eDNA study, But I just wanted to mention that because we've been able to get the workup of tagging a shark, taking our swabs, taking our measurements, taking our sex down to a pretty efficient time period. So it's a lot of work that goes into tagging these sharks and trying to keep it as quick and as safe as possible. So as I mentioned, the PSAT stays on for about 180 days and the splash stays on for about a year. Everything that you see here will eventually grow out of the shark. Yeah, we'll be able to retrieve those PSATs and get all the information from them, which is really cool because if we can retrieve the actual physical device, we're able to download additional information such as accelerometer work, which helps in terms of determining the actual speed of the shark at depth. So I've actually just started my PhD. I'm towards the beginning. I did my confirmation about a month or two ago. And so I haven't gotten to much of my statistical analysis yet, but what we will be using are a couple of different metrics to estimate what the animal's doing without obviously observing the animal. We'll be able to look at probability densities to see where areas of residency might be, and then also using multi-habitat sensitivity models to look at the different how different variables may influence and drive the movement of these animals. And I'm going to try this link. Of course, it didn't work. After I finish the slides, I'll go and show you guys, but I'll show you kind of a live video of what the tracking data looks like. But this is a still video. This was a tiger shark that we tagged up in Harvey Bay in 2022, back in October. We named this boy Fred. He was a 2.7 meter tiger shark. He was tracked for 102 days. And down here, you can see the type of information that we get. So we'll get uplink information, duration of the time it takes for the satellite tag to transfer data to the Argo systems, the amount of satellite passes we get, the length of time it's on. So we tagged this shark October 30th, and it was tagged until February 11th. It was 102 days of tracking data, and then 2 days of satellite transmission. And then this is kind of an idea of what we see with that tracking data. So imagine this shark, it's swimming at depth, potentially down to about 1,000 meters, and we're able to see where it's going, where it's spending more of its time, less of its time. And from this, I'm able to take this information, overlay it with maybe ocean currents, ocean temperatures, seamounts, and from this we can get an idea of what type of activity or characteristics might be defining this type of movement. And then this is what a splash tag looks like. So like I said, the splash tag is near real-time location. So all of the points that we have here would be when the shark was at the surface. This had 200 tracking days at this point in time. So this is for Fred. You can see when the first data point came up, the latest data point, the duration of that. And once again, I can take this and calculate such variables as how long it took the shark to get from one point to another, the type of movement that characterized those travels, and what ocean conditions or environmental variables might correlate with that. That's kind of my next step and what I'm working on now. And those are just the two next to each other. So you can see that we do get a bit of difference in terms of the type of movement that we see between the PSATs and the splash satellite tags. So if you imagine overlaying those, we'll get a better sense of where the shark's actually going at the surface versus below. And then last slide before I go to that link. But essentially, the whole purpose of tracking mature male tiger sharks is to balance the data set. So as I mentioned before, we only have about 3/4 of the population tracking data. We have adult females, and then we have juveniles. But where the males are going is a significant biologically important part of the overall species movement. So being able to target our males, figure out where they're going, should help us determine where potential areas of mating might occur. If we see major differences in males versus female residing areas, maybe we have a pupping ground. We'll get a better sense for when tiger sharks start to make these really long migrations. And essentially, from all of this, we're able to better understand how they interact with different habitats along the coast, their population structure, variation in their movement, trying to determine any population level trends, or if it is just tons of individual variation. Using this, we'll be able to assess the ecological impact of tiger sharks. And then from this, we'll be able to generate better forecasting, policy, bather protection, conservation, and generally provide more up-to-date, effective information to manage this species. So I am going to just exit this real quick and go to that link and show you guys what this data looks like in real time, as it's quite interesting. And so here we go. Right. That's not the one I want. Let me just quickly find the one I want. Where is it? I'll give this one a look. So what's really cool is if I click on this, it takes me in, and this is showing me.
And we're only, sorry, Peter, we're only seeing your presentation.
Oh, are you? Sorry, let me change which screen I'm sharing. So I have to stop and then do a new share. Oh, here we go.
You'll need to stop and share a new one.
Well, what do you see now?
Yeah, that's good.
That's
We know
good.
got a map. Yeah.
It's the Google Earth.
Yeah.
Yeah.
Okay, cool. Awesome. Thank you. So this is my splash tag. So this is the near time locational tag for a shark we tagged out of Coffs Harbor. Green will be towards the beginning of when I tagged this shark back in December, and red is closer to present day. So if we zoom in, not that fast though. Okay. I'm able to play for you guys the movements of this shark. So you can see green when we tagged it down in Coffs Harbor out here. And you can see it heads out, it's moving around the shelf out here, maybe some seamounts. It comes up all the way off of, what is this, Fraser Island, heads out further into the open ocean, comes back along the coast, hangs out. We see a massive travel down here before it heads further north again. And so you can see that we get some very interesting movement here. The next fun step of my PhD is delving deeper into the data here to see if we have anything to do with temperature that's moving this, if there's ocean currents, if they're following prey species. So you can see this shark, he was almost a 4-meter shark. And so this is in July, late July, and August. So you can see that from when I tagged him in December down here off of Coffs Harbour, New South Wales, he's now hanging out up here in the southern GBR in August. So massive movements. And the next part of my PhD is delving into that and kind of seeing what's driving all of that. But yeah, so that's my first chapter. I'll stop sharing my screen. But that's my first chapter of my PhD, looking at that large scale movement. Kind of from that, I'll be able to dive into looking at some more fine scale interactions between tiger sharks and other animals with some of my other tags, as well as some examples and trying to line up what they're eating with where they're moving. So yeah, that's a bit about my project.
Thanks, PJ. That was awesome. If anyone has any questions, feel free to just pop them in the chat. Oh, we've got one already. So from Gabriella, can you give us more details on how you're going to study intra-individual interactions?
Yeah. So we have these really amazing tags. They're called CATS cams, CATS. It stands for Customized Animal Tracking Solutions, if you want to look them up. But essentially, it's a biologging tag with a video camera incorporated into it. So we're able to clamp them onto the dorsal fin of the tiger shark, let the shark go. And what this tag collects is environmental sensors, so temperature, depth, water pressure. And then it also collects inertial measurement units, so acceleration, gyroscope, magnetometer, and then it has the video camera. So we're able to use that to actually record from like 6 a.m. to 6 p.m. or whenever I set it for. The tags are attached to the dorsal fin with the clamp with a GTR, which is a galvanized time release, which essentially means that it's built for X amount of hours. So the GTRs I'm using are built for 24 hours. So after about 24 hours in salt water, they will dissolve. And once they dissolve, the camera detaches from the dorsal fin and floats to the surface. And we go and collect those cameras. And then through analyzing the video footage and the body movements and environmental variables of the tag, we're able to see different interactions between other species, the same species, habitats. I'm able to look at acceleration and see if there was a predation event, if they were getting away from something, going towards something, those type of interactions. So that's how we're going to be looking at that fine scale detail.
Very cool.
Yeah.
So Elena asked if you can show us a video of the camera, but I'm guessing probably not at this stage.
I probably could. So I have 2 deployments that one of my supervisors have done. And I would show you something, but I haven't looked through them all yet. So I would be able to pick a really cool image. But we do have it, and there's a really cool one, not necessarily with tiger sharks, but there's a really cool footage, and Olaf down at Griffith has actually used it in a couple of presentations he's done of whales. And so we hope to get something similar with our tiger sharks, but essentially whales were caught kind of exfoliating themselves on sand, on the ocean floor. And you can actually see the whale like turn over and like rub its back on the sand. And so you get to get some really cool things like that. So hopefully we'll get that with the tiger sharks.
Awesome.
But yeah, I just haven't looked through it yet to pick like a really good video to show.
We'll get everyone to follow you on Facebook or something so they can keep in. So Gabriella just wanted to clarify with the so obviously using those cameras to look at interactions with each individual. She just wanted to clarify whether you can use that still to identify interactions between like pairs of animals.
So
think that's the question.
Yeah, so tiger sharks are solitary animals for the most part, so we're not going to have them in groups. If they are ever found in groups, it's more so because of a big feeding event, such as a whale carcass, but they don't necessarily interact much with each other. Um, so essentially, it will go. Imagine you had like a hat on with a video camera, right? So that's essentially what the video camera is like on the dorsal fin of the shark. So if it turns its head to the right, we'll be able to see what's on the right. If it turns its head to the left, we can see that, kind of what's in front of it. We're trying to decide if we want to put it on backwards one day, so we can get video footage of the back end of the animal and potentially see if there's animals that might move into frame, kind of once it passes, or kind of, the result of them passing through. We'll also be able to collect its tail beat pattern a bit easier that way. But yeah, so I'm not really sure what you mean by like same pair of animals, but essentially, if it goes to, say, predate on a sea turtle, we'll be able to see that motion because it's pointed down towards its head.
Awesome.
And then identify interactions between familiar. So the only way, do you mean familiar to us? Like if we've seen that shark before as scientists, or do you mean familiar to it's the same species, Gabriella? Okay, yeah, so I don't think that we would be there yet. I just don't think our understanding of sharks, large migratory sharks, is anywhere yet where we can identify if they recognize each other in that sense. So no, I don't think we'll be doing anything like that. They don't school tiger sharks, so there's nothing really to look at that way. They're not like dolphins where they recognize specific others and they go and they interact with them and stay with them or choose not to be. I don't think, from what I've read and what I'm familiar with, I don't think particularly tiger sharks are going to have those types of relationships or bonds with other individuals.
Yeah. Awesome. Does anyone else have any questions they might want to ask PJ before we wrap up? I'll just ask one while we're waiting. How many animals do you think you'll tag in total?
Yeah. So the plan is to tag 10 mature adult males. So I don't think I mentioned it, but maturity is determined based on both total length of the shark and calcified claspers. So total length is about 2.7, 2.8 meters. They're close to maturity. And then calcified claspers is essentially claspers on a shark. You grab one in your hand, and if it's nice and firm, it's close to maturity. So the plan is to tag 10 of them, and each one gets both tags. So we're double tagging the sharks with both a PSAT and a splash, so we can get the combination of those two different data tracking types.
Yeah, awesome.
Yeah.
And Tanya's just asking how you catch them / attract them.
We go fishing. So I'm really lucky. I get to work with the New South Wales DPI. So I'll go down to New South Wales and use the smart drum lines that they have. And then if I do my own research stints, we use drum lines and set lines. And you can also just do land-based fishing. So simple hook and rod. And essentially, once you've hooked them, you either reel them into the shore and get them into tonic on their back, or we pull them alongside the boat, and you'll have the two ropes, like the hook rope on one tied up to one side of the boat, and then we put a tail rope on the back end of them and tie it to the other side of the rope, so they'll stay alongside in the water. Tiger sharks are pretty chill. They're really lazy, like they really are. And so they generally do fine hooked up alongside the boat. They don't tend to fidget much or roll too much, which is good for us.
Sweet. And Sam, just to follow up to that, Sam was asking, are there plans for tagging for smart drum lines in Queensland?
So they are testing out smart drum lines in Queensland at the moment up north. If you go onto their page, they do have information there about where it's being tested. It's just one site at the moment. And I believe they have plans to test smart drum lines in a couple other locations. But yeah, it's not as big in Queensland as it is down in New South Wales. That's kind of all I can say on that at the moment.
Any last questions before we wrap up?
Yeah. I'll just ask a question. Do you have many days where you go fishing and you don't catch any?
Yes. Yes. So luckily for us, tiger sharks do have one predictable-ish trait. They do follow a thermocline, and so they like warm waters between like 22, 28 degrees Celsius. And so we do tend to have a tiger shark season, which is end of October to like January, kind of here in Queensland, and then down in New South Wales. It's that summer period again, going even into April. So during that time period is when I focus most of my efforts because we know that they're moving through the area. But I've gone out many a day and not caught a tiger shark, not caught a shark. So it is a bit opportunistic. Next year, I plan on going down south into New South Wales and trying to work with a couple of the competitions that go down there. Since there'll be tons of chum in the water, hoping that helps us get our target species. But yeah, just the nature of working with large sharks, it's kind of hit or miss, and you're hoping that you hit more than you miss. But for me as well, I didn't talk about it here, but my last chapter, which is on diet, we're doing eDNA using cloacal swabs. And we're collecting swabs for any tiger shark that we do catch. So even if I catch a shark that's under the length or maturity level that I'm looking at, I'm still able to collect some data from that individual.
Oh, good. Awesome. Thank you.
I think we had another question from Elena, which was just to clarify how you get the tags back.
Yep. So the PSATs pop up to the surface, and once they're able to connect to the satellite system, we do get a general GPS location. And from that point, if they're close enough to shore and we can get someone on a boat, we can go out and look for it. But we tend to just wait until they wash up on shore, and then hope that someone is in the area that we can ask to go pick it up or look for it. One tag that we had popped off onto Fraser Island, and the rangers were able to collect it for us, which was really lovely of them. So yeah, sometimes we don't get them back. Quite often we don't get them back, but they do transmit most of the data over the satellite, so it's not too much of a worry. It's more of like a big celebration if we do get it back. And then the CATS ones, which are the ones that do more of the fine scale video camera stuff, those pop to the surface. We get a general GPS location, and then we actually have a VHF radio we use to go out and collect it from the ocean.
We've got one from Beverly, and she's asking on behalf of her son what the biggest shark is that you tagged.
Yeah, so I tagged a 3.8 meter tiger shark. And that's the one that I showed you that Google Earth video movement of. He was very big. I've worked with great whites before, and they get really big and they're insane, but tiger sharks are just something else. I love them.
Wow, we keep getting questions. This is awesome. From Sarah, are you correlating any of this to attacks. For example, what kind of movements or sea temperatures, etc., might move tigers into areas of higher human population?
Yes. So a lot of the reasoning behind my study is because tiger sharks are moving further south. And obviously we have tons of surf beaches and, just bather beaches along the coast. So we are trying to figure out more about their behavior to understand if they have triggers, and if they do, what they are, how they could potentially mitigate against them. In terms of the movement, trying to figure out kind of how that we know that they can get really coastal. But we also know those are females. So trying to see if males get coastal as well, looking at then, attacks or instances, whether are more coastal, does that correlate with a particular sex more than the other one? So it's definitely a question we have, but it will come later on after we've looked at both kind of the large scale movement and some more of the fine scale movement that I'll be collecting as well.
Awesome. Great. I think that's pretty much all the questions. If anyone online doesn't mind, I might just quickly get everyone to turn their video cameras on, and I'll take a quick screen share just for all our socials, because it was a really good turnout tonight. If you don't want to, that's absolutely fine. Great. It's almost everyone. Cool. So just go 3, 2, 1 and smile virtually. Great. 3, 2, 1, smile. Great. Thanks, everyone. That's awesome. So if there's no more questions for PJ, everyone's saying good luck with the PhD and for what comes further.
Thank you.
We'll
and if you have questions, email me. It's fine.
Yeah. So if anyone wants to get in touch with PJ, just search up her uni SC profile, or give her a stalk on Facebook or something like that. You can also get in touch with us at Reef Check, and we can pass along any details if you're interested. And yeah, just mark your calendars for the 5th of September, which is when we'll have our next talk. And PJ just popped on that her Instagram is Ocean Alley, if you want to follow her research adventures. I also shared her profile and Reef Check earlier, so it'll be all over our socials. But yeah, with that, I'll just say thanks to PJ again for a really awesome talk, and to all of you for attending tonight. So thanks, everyone.
Thank you.